The outcome, of course, of translation is the production of a protein. But as you probably have seen or have guessed, that as soon as bacteria begin their transcription, translation can get started shortly after. This is different in eukaryotes, but when the messenger RNA is synthesized from the template strand of DNA, as soon as the ribosomes come into contact with it, protein production can begin. And the reason this happens in bacteria is because they don't have a nucleus. So they don't have to wait for the messenger RNA to get out of the nucleus and then encounter the ribosomes, because DNA is located in the cytoplasm of the cell, and so the messenger RNA is going to be readily available to the ribosome. Another thing that bacteria do that we don't see in eukaryotes is that you'll see a number of ribosomes attaching to a messenger RNA molecule, and that is what we call a polyribosomal complex. There is an image of that on the right-hand side of the screen, an electron micrograph. So many ribosomes can be attached to a transcript at any time, so a lot of protein can be made very quickly. This makes sense with bacteria because they do reproduce very quickly. Eukaryotic transcription, of course, is different, and it doesn't occur simultaneously as it does in prokaryotes. It occurs in the nucleus, and then translation occurs in the cytoplasm. The eukaryotic start codon is the same as the prokaryotic start codon at AUG, but in eukaryotes it does not code for formal methionine. Instead, it just codes for methionine. It's still methionine, it's just a different structure to the molecule. Eukaryotic messenger RNA encodes a single protein, unlike bacterial messenger RNA, which encodes many. So in the previous slide, we saw that the ribosomes were busily translating these proteins. Also, bacteria transcribe genes and translate genes in functional units, so there may be more than one gene that is transcribed and translated at a particular time. Eukaryotic DNA also contains intervening sequences of non-coding DNA, which are called introns. And these introns have to be spliced out of the final messenger RNA transcript. And they can be alternatively spliced. So splicing of one sort can yield one protein, and then splicing in another direction can yield another protein. So here we can see this alternative gene splicing in eukaryotes. There are exons and introns. Introns are going to be spliced out, and exons are going to stay. I was a bit confused with that. I always think of exons as being exiting, but no, exons are actually what will stay. Introns are going to be spliced out. This occurs in the nucleus of the eukaryotic cell. An enzyme complexes called spliceosomes are going to form a lariat, or sort of like a hairpin loop, of these introns, and then they're going to be clipped out. So that lariat is excised, the spliceosome is released, and what is left is going to be the exons. And the exons are going to be then stitched together. And now this messenger RNA can leave the nucleus of the cell through the nuclear pores, and then it can be translated. So you can imagine that if the exons and introns are somewhat different here, there can be a different protein completely that would be made. So perhaps this intron is kept and this exon is cut out, and what will result is a protein that is completely different. So this actually was found to be true after the human genome was decoded, because it was found that we have far more proteins than we do have DNA genes that would code for them. So alternative gene splicing explains this. So now we come to assessment question number 10. This one gives students fits. So just try to release your brain. Just think about what proteins could possibly be doing in the cell. We just finished the first two modules, and we learned about all sorts of different proteins that bacteria make. We learned about enzymes such as hyaluronidase. We learned about toxins that bacteria make. We learned about physical structures that bacteria have that are proteins, such as flagella. So for this question, I would like you to come up with a fictitious protein that would be encoded by your gene in number 1. Don't go searching around on the internet for this stuff. You won't find an answer to it. But some of the roles of proteins in the bacterial cell are described on the next two slides. The protein you describe should be fictitious, but it should be based on a factual protein. Now, you're going to have to give consideration as to how this or synthesis of this protein is regulated, and I'm going to discuss operons here in a moment. So you'll have to think about how this gene is going to be transcribed and translated into the protein based upon a particular operon. So there are a couple of choices here. Functional proteins that we find in the bacterial cell include enzymes. So enzymes typically end in the suffix ase. So you can think about some type of a substrate that is going to be broken down by the enzyme, and then you give it a name. It cannot be something that already exists, but it can be something that you make up. It is typically named for the substrate. So if you come up with a fake substrate, the substrate that does something, it has to be based in reality, but it is a name that you're going to make up. You add ase to it, and therefore you would have an enzyme. So a lipase is an example of an existing enzyme that breaks down lipids. Okay. Toxins are also proteins that are found in the bacterial cell. Talked about a lot of toxins in Module 2. There are a number of virulence factors, including flagella, capsules, that are found in Module, that was also discussed in Module 2. And then pigments. You saw that Micrococcus luteus, for example, produces a beautiful yellow pigment, and that pigment is a protein that is synthesized in the cell. Okay. Proteins can also be structural. For example, a capsule. A capsule is also a virulence factor, and capsules may be polypeptide. Most of the time capsules are polysaccharides, but it is possible to have a polypeptide capsule. The cell wall has in it peptidoglycan, and peptidoglycan is a combination of a protein and a carbohydrate. The cell membrane is going to have proteins in it. The cell membrane proteins are for integrity, and they're also going to be for transport. So this could be a protein you're coming up with that transports a molecule into the cell. Flagella and endoflagella are made of proteins, knowing that they are made of a protein called flagellin. Ribosomes have proteins associated with them. DNA has proteins associated with it. Those are the non-histone proteins that help in supercoiling the DNA. And then surrounding the spore, the endospore, we can also find proteins making up the coat. Okay. So gene expression is the production of a protein. We say when a gene is expressed, then a protein is going to be synthesized. Sometimes gene expression is not regulated, but most of the time it is. It's highly regulated in eukaryotic cells and is very complicated. In bacterial cells, not so much. It's not as complicated, but it can be regulated. Constitutive enzymes are enzymes that are always produced by cells independently of the composition of the medium in which the cells are grown. So constitutive enzymes are on all the time, and the gene to make a constitutive enzyme is called a constitutive gene. And an example of this would be the enzymes that operate during glycolysis and the tricarboxylic acid cycle, or the Krebs cycle. These are generally constitutive enzymes. They're present at more or less the same concentration in cells at all times. So the genes that code for these enzymes, these proteins, are always turned on. There is no regulatory mechanism that shuts them off. Others are expressed only when necessary. For example, an inducible enzyme will be produced or turned on in cells in response to a particular substrate. They're only produced when needed. In the process of induction, the substrate, or a compound similar to it, is going to evoke the formation of the enzyme, and we call that an inducer. We're going to see an example here of an operon, but an inducible enzyme is only going to be made when the substrate for that enzyme is present. So the gene to make that enzyme is going to be turned on when the substrate is present. When the substrate is not present, the gene is going to be turned off. A repressible enzyme, on the other hand, is one whose synthesis is downregulated or turned off by the presence of the end product of a pathway. So in this case, the end product is called a co-repressor. A repressible enzyme is one that is on all of the time until it is turned off by the presence of the end product of the pathway. So we're going to take a look now at two different operons, the inducible operon and the repressible operon, that are involved in gene expression of the bacterial cell. Okay. Now, before we get to that, there is non-genetic regulation of protein synthesis, and this is the same thing that happens in eukaryotic cells. It's called feedback inhibition, and this is not a genetic regulation. In feedback inhibition, the end product of a biosynthetic pathway inhibits the activity of the first enzyme that is unique to the pathway. So cells will not make product wastefully. The first enzyme in the pathway is an allosteric enzyme. That means it has two sites. One is the active site, the other one is the allosteric site. The end product of the pathway is going to bind to the first enzyme in the pathway and is going to shut it down temporarily. Let's take a look at a video on that.
Many of the enzyme-catalyzed reactions that occur in a cell, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, the product of one reaction becomes the substrate for the next reaction. If the end product of a pathway, such as an amino acid, becomes available in the environment, it is unnecessary and wasteful for the cells to continue to produce the product. Cells therefore have the ability to shut down a pathway when it is not needed. In feedback inhibition, the end product of the pathway reacts with the first enzyme that is unique to the pathway. The reaction occurs at a site on the enzyme that is different from the active site, called the allosteric site. When the product binds to the allosteric site, the enzyme undergoes a conformational change and can no longer react with its substrate. There is no substrate for subsequent steps in the pathway, and the final product is no longer synthesized.
So for assessment question number 11, you're going to describe the regulatory mechanism that governs the transcription of this fictitious gene that you have in number 1. You want to include drawings illustrating the operon in both the on and the off modes. So you're going to model your drawings after these slides that you're about to see. For this question, you have two choices. You can choose an inducible operon or a repressible operon. As a model, then, you're going to be using the Lac Operon as your inducible operon or the Trip operon as a repressible operon. This is all going to be based on the protein you invented in question number 9. Now, an operon consists of several genes that we're about to see, so your gene will be one of them. Remember, this is fictitious, so you can use creative license when you create this. Okay. So operons are coordinated sets of genes, all of which are regulated as a single unit. There are two types, inducible operons and repressible operons. Inducible operons are induced or turned on by the substrate for the enzymes that are synthesized as a result of gene transcription and translation. Inducible operons are usually involved in catabolism. In other words, the enzymes that are synthesized by the inducible operon are usually involved in the breakdown of molecules. On the other hand, repressible operons are also genes in the series, but they are turned off by the product synthesized. So typically, these are used in anabolism. As the product builds up, product is being made. If the product builds up, then it is going to shut off the operon. Operons are made of 3 DNA segments, and as far as an inducible operon, the Lac Operon is the best known. So we're going to take a look at that as the model. Your question then, number 11, is going to be either modeling the Lac Operon or modeling the Trip operon. There will be 3 DNA segments. The first is the regulator gene. The regulator gene codes for a protein that is known as the repressor protein. The repressor protein is allosteric. It has one binding site for the operator region of DNA and one for the inducer molecule. So you remember the operator region. The operator region is where RNA polymerase binds to make messenger RNA. The repressor gene on the chromosome is going to be a distance away from the operon. So it's not contiguous, it's not right next to the operon, but it is at another site on the circular chromosome. The repressor protein is going to be produced constitutively. That means it's being made all of the time. It doesn't turn off and turn on, it's made all of the time. There's also a control locus. The control locus is composed of the promoter region and the operator site. The promoter region, remember, binds the RNA polymerase. And then the third component is a structural locus. A structural locus is made of a number of genes, each coding for an enzyme. Now, in the case of the Lac Operon, there are 3 genes, and each of those 3 genes is necessary to catabolize or break down lactose. Remember, inducible operons are typically catabolic. So this one has 3 genes. Perhaps the one that you describe will only have 2 genes, or you can have it have 3. And again, you can make up the names of these genes and their functions. So the 3 genes that are involved in the Lac Operon include the LAC-Z. That gene codes for beta-galactosidase. Beta-galactosidase is an enzyme that hydrolyzes lactose or breaks down lactose. The LAC-Y gene codes for an enzyme called a permease. Permease brings lactose across the cell membrane. And then finally, the LAC-A gene codes for beta-galactosidase transacetylase, and exactly what that does has not been figured out yet. The Lac Operon will only function in the absence of glucose. So if the cell does not have any glucose available to it, the Lac Operon can be turned on. If there's lactose and glucose present in a cell, the cell will continually use glucose. It will not use lactose. Once the glucose runs out, then the Lac Operon can be used. Now, the Lac Operon is an inducible operon, which means it is normally off. If there's no lactose around, or if there's glucose around, the repressor protein binds to the operator locus and blocks transcription of the downstream structural genes. Okay? So this repressor protein actually docks onto the DNA, and it acts like a boulder that is blocking the operator locus. So RNA polymerase can't bind and can't do its thing. If lactose is available, that's the inducer molecule, it's going to turn the operon on by binding to the repressor protein. When lactose binds to the repressor protein, it changes its shape and causes it to fall off the operator. This repressor protein now falls off the DNA. RNA polymerase can bind to the promoter region, and the structural genes all can be transcribed. The structural genes will be transcribed to those 3 proteins I talked about, and then more lactose can be brought into the cell, and lactose can be degraded. Okay. So this is an image that you can model what you are drawing on question 11. And I'm going to show you a video of this so you can more easily understand what's going on. Right? So remember that there is the operator region. Here we have those 3 genes. So those are the 3 structural genes. And then there is also the LACT-L gene. That is the gene that is coding for the repressor protein. Okay. So the repressor protein looks like this. The repressor protein is capable of binding to the operator site, and that blocks RNA polymerase. When lactose is available, lactose will bind to this repressor protein, and it changes its shape. When its shape is changed, it is incapable of binding onto the DNA, and therefore RNA polymerase is not going to be blocked. Let's take a look at the video.
The E. coli Lac Operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the Lac Operon: beta-galactosidase and permease. Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose is the Lac Operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 Lac Operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA, and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the Lac Operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
Okay, so that's an example of the Lac Operon, and the Lac Operon, you can see, has the regions that are described here in this slide. So review this information, and then you are going to base your question number 11 either on the Lac Operon, or, which is an inducible operon, or on the trip operon. The trip is a repressible operon. That should be an I there, not repressible. It should be repressible. Sorry about that. And it's made of 3 DNA segments as well. So there's also a regulator. The regulator is a gene that codes for a repressor protein, but in this case, the repressor protein is going to act a little bit differently. The repressor protein is also allosteric. It has one site that binds to the operator region and one for the inducer molecule. The repressor gene is on the chromosome a distance away from the operon, just like it is in the inducible operon. And the repressor protein is also produced all of the time, constitutively. There's a control locus. The control locus is composed of the promoter and the operator sites, and the promoter region binds RNA polymerase. Then there's the structural locus, and in the trip operon, there are 5 genes. These 5 genes are each going to code for an enzyme that is needed to anabolize or synthesize tryptophan. That's why it's called the TRP, or the trip operon. TRP is the abbreviation for tryptophan. The trip operon also has an attenuating region that further controls the speed of tryptophan production. This is not something that you have to be concerned with your assessment, but just something that happens with the trip operon. And tryptophan is referred to as a co-repressor in the operon's functions. So in the Lac Operon, which is an inducible operon, lactose is the inducer. Here, tryptophan, which is the product of the reaction that is going to happen becomes a co-repressor in the operon's function. Okay. So the trip operon is normally on. So it's on all of the time, and tryptophan is going to be made. Tryptophan's an amino acid, so it's something the cell is going to need. It needs to make proteins. So tryptophan is made, tryptophan is used up, and at some point, perhaps the cell is at hard times. It finds itself in a terrible environment. Reproduction is not going to continue, and tryptophan builds up. Tryptophan, which is the co-repressor, turns the operon off. Now, if you're following along with the notes, there was an error there. It says that it turns the operon on. It does not. It turns it off. Binding of tryptophan to the repressor protein's allosteric site changes its shape. So just like with the Lac Operon, the shape is changed. But in this case, it allows the repressor protein to bind to the operator locus. In the Lac Operon, it fell off. So now it's going to bind on. Now it will block RNA polymerase because it's like a boulder sitting on top of the DNA. RNA polymerase's access to the structural genes is now blocked, and the structural genes are not transcribed. There'll be excess tryptophan. When tryptophan gets used up now, the repressor protein is going to fall off, and now the operon is going to be turned on. So here again, we see that the locus that codes for the repressor protein. The repressor protein has this particular shape. When it binds to the co-repressor, it changes shape, and when it binds to the co-repressor now, it can bind to the operator locus. So in this case, when tryptophan is present, the repressor is bound to the operator, the operon is repressed. When tryptophan is absent from the environment, then we're going to have that initial shape of the repressor protein. Whoops, excuse me. And in the absence of tryptophan, the free trip repressor can't bind to the operator site, and RNA polymerase can move past it and transcribe the trip operon genes. Now the cell has the capability to synthesize tryptophan. So you can see there are quite a bit of enzymes that are being involved here, and certainly not as much as you would have to consider for your assessment. But for your assessment, you are either going to be considering an inducible operon or a repressible operon. So let's take a look at the video that describes the trip operon.
The tryptophan operon, trip operon for short, is a bacterial system for regulating the synthesis of the amino acid tryptophan. The operon controls expression of 5 genes. These structural genes are transcribed when RNA polymerase can bind the promoter controlling them. But when the cell has lots of tryptophan, why waste energy making more? The trip operon is at one position on the circular bacterial chromosome, and a regulatory gene for the system is about a quarter of the way around the chromosome from it. This gene, called TRPR, encodes a dimeric repressor protein on a slow but ongoing basis, and the repressors diffuse through the cytoplasm. We'll see in a little bit how the repressors work. Now let's look at the control sequences for the trip operon. They include a promoter, the binding site for RNA polymerase, an operator, which overlaps the promoter, and a leader gene, TRPL, which has an attenuator sequence, which we'll also look at soon. When the bacterial cell's tryptophan levels are low, RNA polymerase can bind the promoter and transcribe the structural genes. Each gene encodes a protein chain for an enzyme in the TRP synthesis pathway. TRPE and D for anthranilate synthase, TRPC for indole glycerol phosphate synthase, and TRPB and A for tryptophan synthase. As tryptophan is synthesized, it diffuses through the cytoplasm. When it encounters a trip repressor, it binds a site on the repressor, changing that protein shape. The repressor can now bind the operator, block polymerase binding, and prevent transcription of the structural genes. Thus, tryptophan is said to be a co-repressor. Like the Lac repressor, the trip repressor does not stick like glue, but periodically dissociates allowing a low basal rate of transcription. The operon can fine-tune tryptophan synthesis. In essence, it can read various levels of tryptophan concentration in the cell. It uses a system called attenuation to do this. When the tryptophan level is low, ribosomes translating the leader mRNA as it emerges from the RNA polymerase pause briefly when they encounter two tryptophan codons in a region called 1. They wait for trip-charged tRNAs in order to continue. And while they wait, a pair of complementary regions of the mRNA, called 2 and 3, form a stem and loop. The next region that could have paired with 3 is not yet transcribed. When the trip tRNAs appear, translation continues through the structural genes, and thus more tryptophan is produced. If, however, tryptophan-charged tRNAs are plentiful, there is lots of tryptophan in the cell, the ribosomes read through region 1 quickly, which allows region 3 and newly transcribed 4 to pair. This stem and loop terminates transcription, and the structural genes are not transcribed. Tryptophan is not produced, and its concentration in the cell decreases.
Okay, so I don't expect you to include anything about attenuation there. You are just going to simply discuss the transcription and translation of the structural genes. Okay, stop with this one and we'll continue the recordings for question 12.